today we're going to ask the question, can CD8 T-cells be harnessed to eliminate latent HIV reservoirs? And to start off, I want to briefly review the evidence that CD8 T-cells play an important role in controlling untreated infection. And you heard some of this earlier this morning as well. So the first pillar of evidence, if you will, that CTLs play a role in controlling HIV is that in early infection, if we look at HIV viral load in red and CTLs in purple, the emergence of the HIV-specific CD8 response coincides with when the viral load is brought under partial control. So they're in the right place, or they're present at the right time to be mediating some effect. The second pillar of evidence is that people with certain HLA-B types tend to have a better ability to control infection. And of course, the role of these HLA-B molecules is to present viral antigens to the immune system. So this seems to implicate CD8 T cells in different levels of viral control. Third, from animal models, which we've also heard about today, if you deplete CD8 T cells, you see a dramatic increase in the viral load. And finally, of course, if we just take HIV-specific CD8 cells in vitro, we see that they very effectively kill HIV-infected cells. So it makes sense that they can play a role in natural infection. And I'm sure you've seen movies like this before, but here's one we took with an HIV-specific T cell encountering its target and killing it very dramatically. So we can see this very clearly happening in vitro. So why doesn't this clear infection in vivo? How come antiretroviral therapy, along with this CTL response, isn't enough to cure infection? This was a review from just a few years ago that I wrote with Bruce Walker, highlighting what we thought were the main barriers preventing the CD8 cells from clearing infection. First of all, there's issues with sequence diversity and immune escape. So if the CTLs can't recognize the virus in the reservoir, then of course they can't kill it. The second is T-cell exhaustion or dysfunction. CD8 cells progressively lose the ability to proliferate, to make cytokines, and to kill cells. We also need to think about compartmentalization in people. Certain places like lymph node follicles can harbor high levels of infected cells, but are poorly accessed by cytotoxic T cells. And the last barrier, which I'm going to spend most of today talking about, is HIV latency. So if the virus is in a state where it's not presenting antigens, then the CD8 T cell is not going to be able to recognize it. So there's different strategies that people are exploring to try to overcome those barriers. And what we proposed, and what I think most people think is the case, is that if we can overcome these 4 barriers, then maybe we can eliminate or at least reduce the viral reservoir. So we're going to focus on HIV latency. So we'll get to that in a bit, and I'm going to talk today mostly about our efforts to try to eliminate that viral reservoir in vitro. But first, a bit of a background as to what is a latent HIV reservoir. So in infection, HIV generally activates or infects activated CD4 cells, most of which are killed by the virus or by the immune system. But some of them seem to survive this process and return to a resting state. And then these can be long-lived, latently infected cells which aren't exposed to the immune system. And we can measure these cells in people in different ways. So people who are on long-term therapy. We can take CD4 cells from a person, plate them in wells with a different number of cells per well, maximally stimulate them to get them to produce virus. And at the end of 14 days, we just count up the number of wells that have infected cells, and we can calculate the frequency of the patient cells that had an infectious virus. And this is called a quantitative viral outgrowth, or QVOA, assay. So one of the early signs that cure wasn't going to be easy came from studies from the Silicano lab, where they used this assay and studied people longitudinally over several years and asked whether this latent reservoir decays on its own over time. And unfortunately, the result of this study, which has subsequently been reproduced by the Margolis lab, is that the rate of decay is very, very slow. So it'll take more than a lifetime of therapy for the viral reservoir to go away on its own. So we need to do something more than wait. We need to somehow purge this HIV reservoir. So now we're going to kind of sequentially go through the stages that I think the field went through when they thought through this problem. At first, it was thought if we can just reverse latency, then the virus can probably kill the infected cell, and this will be enough to reduce the reservoir. Makes sense. The virus is pretty cytopathic. So that's the idea. And this actually works pretty well, usually, if we have a strong latency reversing agent in a primary cell model of latency. So what do I mean by a primary cell model of latency? I have to introduce that concept before we can talk a bit more about the results. So there's several different models out there. This is what we call the post-activation latency model, such as the one that the Planellis and Bosque labs use. They activate CD4 cells, infect with HIV. This is in vitro. They can then start therapy, and over the course of days to weeks, some of the cells are killed, but some reverse to a resting state. And then the idea is that these cells resemble the latent reservoir that's in patients. And again, in this model, if you have a strong latency reversing agent like bryostatin, like C3 or CD28, then these cells actually do die when they're reactivated. But I need to make a distinction here, which is going to be important throughout the talk, between these primary cell models of latency and the real, what I'm going to call the natural reservoir. And the natural reservoir are infected cells that are in CD4 cells that are taken directly out of someone living with HIV, so someone who's on long-term therapy. And a landmark paper from the field was again by the Silicano lab, by Liang Shan in 2012, where they just asked the question, if they take CD4 cells, very large numbers of CD4 cells, because the latent reservoir is very infrequent, treat these with a latency reversing agent called SAHA, or with a control in vitro, and then use this QVOA assay that I just described to measure whether this actually reduced the reservoir. By waking up the virus, did we kill these infected cells? And this was the result. This is without treatment, this is with SAHA treatment, and there is no difference in this reservoir. So this was kind of a discouraging result that suggested that something other than latency reversal, at least with this mild latency reversing agent, would be needed to clear the reservoir in these real patient CD4 cells. So this really gave some traction to the idea that we need to involve the immune system, and HIV-specific CD8 T-cells, or CTLs, are one type of effector we can use. Natural killer cells, antibody toxins, there's a number of different approaches that could be taken. But this gives rise to this idea of kick and kill, or shock and kill, which are essentially the same thing. So now, in addition to just reversing latency, we're going to bring in an HIV-specific T-cell that will kill this reactivated cell. Okay, so the LRA exposes the virus, and then the CTL kills the cell. And we'll go back to this same Liang Shan paper, where they started to test cytotoxic T-cells. But a point I want to make here, which is going to be also important for the direction that we go in this talk, is that now in this paper they switched from these real ex vivo cells from patients, so from natural HIV reservoirs, to one of these latency models that I also talked about. So if you use one of these latency models, where it's a short-term infection in vitro and let it go back to resting, if you reactivate these cells, again with SAHA, and then add expanded CD8 T-cells, so each of the dotted lines here are from different HIV-infected individuals with CD8 T-cells that have been expanded. Here we can decrease the number of cells that are expressing GFP. So they started off showing that latency reversal alone wasn't enough against the patient reservoir, and then they show that latency reversal plus CTLs are good against a primary cell model. The direction that we're going to go in is to ask if cytotoxic T cells and latency reversing agents can actually kill the real natural reservoir from people with HIV. That's just what I said here. So these are results from latency models. Does this actually work against infected cells from patients, which may have some features that are not present in the latency models? Another thing to point out here is that these experiments used a NEF-deleted virus in this model. And remember, NEF is important for immunoevasion by downregulating MHC1, so that might also be important. So an important step for the field in the right direction, but I think leaving some additional experiments to do to really validate the kick and kill principle. So this brings us to what my lab is mostly interested in. Most of the validation of shock and kill concepts come from model systems. If we take CD8 T-cells with latency reversing agents, can we actually drive reductions in CD4 cells immediately out of people living with HIV? Okay, so I'm going to present results from these experiments in the latter part of the talk. Just before getting into that, I want to do a bit of an overview of some of the complexities of the reservoirs, because we're going to need these to understand some of the aspects of our results. Okay, so I mentioned one way that we can measure the viral reservoir using a QVOA assay. However, we can also measure HIV-infected cells using HIV DNA, using a real-time PCR method. And when we apply this to our cohorts, we actually see that there's, in our hands, a nice correlation between the frequency of cells that have an infectious virus, as measured by QVOA, and the cells carrying HIV DNA. But you can see if you trace this across, there's about 1,000 times more cells that have HIV DNA than the cells that measure positive in our viral outgrowth assays. And the reason is that most of this is from defective proviruses. You'll notice here that we have some nice cohorts, not only from the US and also from Canada, but also from Brazil. And this was in collaboration with Camilla, and she came up and visited our lab, and we did these experiments together. So a bit of a aside is also something we noticed when doing these experiments is if we measure the actual reservoir sizes, we see that the highest reservoirs here are in our American samples, followed by Canada, and the lowest infectious reservoirs are actually from Brazil. So it's America first here, at least for the reservoir. Okay, so a lot of these cells are defective proviruses. What does this actually look like? This is a nice study from Katie Bruner, where she just does very time-consuming and difficult PCR assays to characterize the proviruses in these cells. This is from her Nature Medicine paper. You can see the blue shows the sequence that's intact, and this white is all holes in the provirus. So if you start to sequence these viruses, it's actually really hard to find an intact HIV provirus. You have to look through a lot of these defective proviruses. And by their characterization, only about 2% of cells actually had intact proviruses. Okay, but again, this is leading to our assays where we try the shock and kill in ex vivo samples. But before getting to that, we need to know how to think about these defective proviruses versus cells with infectious proviruses. And we first ask the question, are these defective HIV proviruses really inert? Are they really never expressed? Are they junk DNA? And to ask this, we teamed up with Yachi Ho and published this paper last year showing that some of these defective HIV proviruses can actually be expressed and can be recognized by cytotoxic T-cells, which can shape the proviral landscape. So maybe we can't haul them off to the junkyard just yet. The initial result that made us think about this came from Yachi's work. It's hard to see some of the details here, but this is just sequencing RNA that was being expressed in ex vivo CD4 cells. And she noticed that in some of these individuals, quite a bit of this RNA, which is in green here, is hypermutated proviruses. And this is one type of defective HIV provirus. So she asked the question, can these hypermutated proviruses actually be expressed? They're unlikely to make normal proteins because they have stop mutations and other defects in them, but maybe they can be presented to HIV-specific T-cells. So to test this, we designed an assay where we use CD8 T-cells as sensors to ask the question if they can recognize a cell with a defective provirus. So Yachi had cells that she transduced with defective proviruses from actual patient cells. These are real clinical samples. And then we mixed them with our HIV-specific clones and asked whether these responded by degranulating. Here's first our negative control, which is a CMV-specific clone, which doesn't respond to any of these. These are all different defective proviruses, but of course does respond to the peptide. This is a pretty complex figure, so it's in the cell-hosted microbe paper if you want to see details. I just want to draw attention to a few things. In red here, this is one type of defective provirus. This is a hypermutated HIV provirus. So even though this can never produce an infectious virus, it probably can't produce even an intact protein. When we mix it with HIV-specific CTL clones, the clones respond. So the clones can still see these cells that have defective proviruses. Some of the other defective proviruses are not recognized, those that have deletions that span the epitope, for example. So it's a bit of a complex story. Not all defective proviruses can be recognized, of course, but some can. So the analogy for the HIV reservoir that I'm going to present, and I'm going to use it throughout the rest of the talk, is that maybe we can think of it a bit like a beehive. So in the beehive, only the queen can reproduce, right? The queen's a small minority of the bees. And I'm going to say that this is like the cells with the infectious HIV provirus. Only those cells can reestablish viral replication and infection. But that doesn't mean that we can forget about all the other bees, the defective, the replication incompetent bees. And that might also be the case if some of these defective proviruses can stimulate the immune system. So throughout the rest of the talk, there's going to be a lot of figures. So when I am showing something that represents the intact, infectious reservoir, I'm going to show the queen bee next to it. And the defective provirus, I'll label it with the drone bee. So please remember this part, otherwise the insects will be confusing in the next slide. Okay, so what latency reversing agents do we want to use for these assays? So one thing about seeing if we can shock and kill CD4 cells from patients is that because the frequency of infected cells is very low, around 1 in a million, we need to use a lot of cells for these experiments. We typically use 5 billion cells in an experiment. So before doing this, we need to pick which latency reversing agents we think are going to be best. And there's a number of different agents that can wake up HIV, reverse latency. And I've arranged them here in what I would call increasing effectiveness, but decreasing safety. So at the top, we have some relatively safe agents, but they may not do as good of a job of reversing latency from patient samples. And at the bottom, we have things like anti-CD3s, so very strong agents for waking up the virus, but not things that we want to give to people who are living healthy lives on antiretroviral therapy. So what do we need for a latency reversing agent? It has to both induce HIV antigen expression, of course. So we don't want to necessarily just induce some HIV RNA expression. We want to make sure that that's resulting in presentation of actual antigens on MHC1, something that the T cell can see. And the second requirement is it's better if we give an agent that does not impair cytotoxic T cell function. And some of the HDAC inhibitors, at least in an in vitro setting, can impair some of those CTL functions. Okay, so to ask which latency reversing agents we want to use, we come back to this system. We're going to treat the latently infected cells with LRAs, mixed with the CTL, but now we're going to measure cytokine production and degranulation from the CTL itself. So this is the sensor to see if the latency reversing agent worked. And here's an example of an experiment. In the top, we have an HIV-specific clone. And we can see that where we treated the latently infected cells with IL-15, the CTL clone was then able to respond to these infected cells by degranulating, or sorry, upregulating CD137. IL-2 was also effective in this assay. We did not see the induction of antigen presentation with this HDAC inhibitor, SAHA. This protein kinase C agonist prostratin did also induce antigen expression. This is a CMV-specific clone, which is just an irrelevant control. So here's just compiled results using a number of different CTL clones. We can again see that IL-15, PKC agonists, IL-2, and TLR2 agonists were inducing antigen expression from the resting cells, whereas, again, in our hands in these assays, the HDAC inhibitors weren't giving us the readout that we were hoping for. So we're not going to focus on those. Okay, and just to mention again, as we move into these ex vivo assays, we're really dependent upon leukapheresis samples, which get large number of cells from patients. So we have really dedicated study participants and clinicians that we're grateful to. Okay, so coming back to this main question now with that extra background, can combinations of CD8 cells and latency reversing agents really drive reductions in CD4 cells? Which CD8 T-cells do we want to use for these assays? We teamed up with Kath Bollard's lab, and we're using a type of actual clinical grade HIV-specific T-cell products. So these are enhanced CD8 T-cells that are very similar to products that she infuses into patients for other types of viral infections. So we can take T-cells, mix them with peptide pulse dendritic cells to expand up the HIV-specific cells. And then they get a number of treatments with different cytokines like IL-7 and IL-15, as well as some feeder cells that express some co-stimulatory molecules like 4-1BB ligand, CD80, 86, to really make them as optimally effective as we think that we can. These products that she generates kill very well. Peptide pulse cells in vitro. This is just a chromium release assay, and these are all the peptide pulsed versus unpulsed targets. We're also starting to gather some evidence that these T-cell products can actually suppress HIV replication in mice. So these are NSG mice that are engrafted with human CD4 cells and infected with JRCSF. And these are some preliminary data, but it's clear that when we give the HIV-specific T-cell product, we can suppress viral replication in these mice. So the T-cell effectors look good. We think we know which latency reversing agents we want to use, so we have those two pieces of the puzzle, and we have some background into what we're going to measure in terms of defective and intact proviruses. So this is now the kind of the meat of the assay. So to test whether we can reduce these reservoirs, we developed something that we call the HIV eradication assay, or short form is the HIVE assay. So we start with ex vivo CD4 cells from people on long-term therapy. We treat with different combinations of latency reversing agents, with or without our effectors here, so our HIV-specific CD8 cells. We give them 4 days to try to shock and kill these latently infected cells. We have antiretrovirals to stop any spread of infection. And then after these 4 days, we deplete the CD8 cells, and we have pure populations of CD4 cells that have either been exposed to kick and kill strategies or have not. Not quite done yet. Then we take those cells and we do a battery of tests on them. We look at viability and cell counts. Some of these cells go to look for HIV DNA, which will be total infected cells, defective and intact. And some go into our QVOA assays to see if we can have actually depleted the replication-competent virus. And this is, I think, a very important step. Okay, so what results do we get with these nice clinical-grade T-cell products, and starting first with a strong latency-reversing agent, bryostatin-1, which is a PKC agonist. When we look at HIV DNA, which is largely defective provirus, as we remember, we see that the combinations here with either an IL-15 superagonist and a CD8 cell, or bryostatin and a CD8 cell, saw significant reductions in HIV DNA relative to the no treatment or the LRAs only. So that was very encouraging. Unfortunately, when we looked at the viral outgrowth assay that measures the replication-competent virus in the same assay, we were surprised to see no difference between these different conditions. And so this is, I think, makes sense really also only in the context that some of these defective proviruses can be recognized by CD8 cells, which we showed earlier. So we did a number of these experiments. I'm going to show summary data later with these clinical T-cell products and saw similar results. One of the explanations could simply be immune escape. So maybe a large portion of the defective proviruses are not escaped from the T cells, but the intact virus might be largely escaped. So we need to know really what our CTLs are targeting. So to do this, we switched to HIV-specific T-cell clones, and these are isolated from blood of individuals living with HIV, and it's the population of cells that are all specific for one single epitope. So we know exactly what they're targeting and what they can kill. I think the movie's not going to work. It's similar to the one I showed before, but this is just a killing assay with productively infected cells. So not the latent reservoir now, just cells that we've newly infected with HIV. And we see that the clones kill these very nicely. So we take these now into our HIV eradication or HIVE assay. Here are the 3 clones with their peptides, just showing that they are specific. Here they're degranulating in response to peptide. In HIV DNA, we see the similar pattern that I showed you earlier, where we have combinations of the clones with the latency reversing agent. We have reduced the amount of HIV DNA, and these results are part of a study that we published in JCI recently. However, again, when we did this viral outgrowth assay, we were disappointed to see that there was no difference in all of these different conditions. Because we know exactly what this clone is recognizing, we can sequence the virus that grows out of these wells and ask whether it has escaped from these 3 different types of CTL clones. The ones that were treated with this clone were largely escaped, 71%, but the other 2 types of clones, the virus did not escape from it, so we can't attribute it to immune escape. Just to be sure that the virus hadn't escaped, so when you do these QVOAs, although the main point is to measure the reservoir, at the end you have a well that has virus in it, the same virus that you just tried to kill. So you can take that virus, you can infect activated cells, you can mix it with the same CTL clone that you used in the HIV eradication assay, and ask whether the clone responds by producing CD107A, so by degranulating. And what we saw in this experiment, this is the negative control. Here's all the supernatants from the QVOA. Here are the positive controls, is that the clone could still recognize these wells with virus. So even though it didn't kill them in this HIV eradication assay, when we took the virus and infect new cells, the clone could see and kill those newly infected cells. So we started to get more aggressive, more desperate maybe with these assays. We got away from things that might be able to go into the clinic, and we just said, what if we take the strongest latency reversing agents that exist? In this case, PMA ionomycin, or here, anti-C3 CD28. When we add these agents, again, at the DNA, which is largely defective, we are seeing significant reductions. Here, the LRA alone also induces some reduction, but this might be some proliferation of uninfected cells. When we do the QVOA, we again see a lack of a decrease in these experiments. So the story is a bit disappointing, but consistent at least. And we can summarize it here. So these are all the experiments we've done. It's a bit of a mixture of different latency reversing agents. We're just trying to take our best shot every time. Sometimes the latency reversing agents increase the HIV DNA. Sometimes they decrease it. But when we add the CTLs, we always see a nice decrease from the latency reversing agent alone. When we look at the infectious virus, so the queen bee, the one that can actually reseed infection, we can increase the reservoir with the latency reversing agent, but we can't seem to drive it down here when we add our different types of CTL clones. And this is very different from what we saw and what others have seen in latency models. So what it seems to be telling us is that this real reservoir from people living with HIV seems to resist elimination in a way that we haven't seen with latency models. So bringing us back to this summary figure that I showed you earlier, we feel that we overcame all of these obstacles that we had envisioned. We picked our clones carefully to know that the virus had not escaped. The clones that we picked were highly functional. They were able to kill productively infected cells, at least. There's no issue of compartmentalization in our in vitro experiments here. And we definitely did our best to overcome HIV latency with agents that were much too strong for the clinic. And yet we didn't seem to eliminate this reservoir. Another way to summarize this is from Ya-Chi Ho's paper, her iconic representation of the reservoir. Some of the defective proviruses don't express antigens and can't be recognized by CTLs, but some do seem to be recognized by CTLs and can be eliminated. But we weren't able to eliminate these intact inducible reservoirs. But what's really interesting is if we take the virus that grew out of these cells and infect new cells, the CTLs can kill those. So it seems to me that there's something about these real cells harboring the virus. Remember, these cells have lived for years or for decades in these people, where it may not be as simple as inducing antigen expression and that the CTL can consistently kill these. So we might need something else for this shock and kill approach. Can we do something to sensitize these target cells that have the replication competent virus so that the CTLs can kill them? And our attention was drawn to this paper from Andrew Badley's group, which presented data that a BCL2 antagonist was able to increase the clearance of latently infected cells in their system. This didn't involve CTLs, not a shock and kill, just a shock with the BCL2 antagonist. So could this work? Could this help CTLs kill infected cells? We weren't really sure, but here's at least a plausible mechanism by which this might work. So when a CTL recognizes a target cell, perforin forms a hole in the target cell through which granzyme can pass, and granzyme acts directly on caspase 3. This is how most people think of CTL happening. This is the dominant way that CTL killing occurs. But it can also cleave this molecule BID to TBID, which can induce mitochondrial death. And BCL2 proteins directly antagonize this mechanism of death. So there's at least a plausible mechanism by which a BCL2 inhibitor could enhance killing of target cells. So let's try it. And to try it, we shifted away from the CTL clones, and we shifted back to these clinical grade T-cell products that I introduced to you before. And here's the first experiment, and it's the first time we actually saw a reduction in the QVOA, so it was an exciting moment for our lab. Here's the infectious reservoir. CD3, CD28, a very strong latency reversing agent. ABT199 is the BCL2 inhibitor, so those agents alone aren't decreasing the reservoir. The CD8 T-cells plus these are not decreasing the reservoir, but where we added this triple combination, now we had no positive wells left and a highly statistically significant reduction in the reservoir in this first experiment. So we've expanded this out now to a number of different participants. I'm showing first the results with the total HIV DNA. And again, with latency reversing agent alone, we don't decrease HIV DNA, this defective provirus, or with the latency reversing agent and the BCL2 inhibitor. As we've seen before, these are completely different experiments than I showed you earlier and than what we published in JCI. But consistently, this latency reversing agent with the CD8 T-cell reduces HIV DNA slightly, even though most of this is defective. And it looks pretty similar where we add the BCL2 inhibitor, not a big difference. Where we see the difference is with this infectious reservoir, the queen bee. So here, the latency reversing agent alone doesn't decrease the reservoir. The latency reversing agent plus the BCL2 inhibitor, we do see actually that sometimes we see reductions in the infectious reservoir, but sometimes we don't. And over the population, it's not significant. As we reported before, the latency reversing agent and the CD8 T-cell alone usually doesn't decrease the reservoir. In one case, it did. So we're studying what's special maybe about this CD8 T-cell, but generally what we haven't seen. But this triple combination, latency reversing agent, HIV-specific T-cell, BCL2 inhibitor, now we're seeing at least partial reductions in this infectious reservoir. In some, it goes to 0. In some, we still have some ways to go. So one more type of experiment to try to enforce this point, and we're doing more of these, but I want to introduce something we call the spiked HIV eradication assay. So I hope that one of the points that I've made is that we think there's something different about the natural HIV reservoir, so these cells that are actually coming out of people living with HIV versus these latency models, which are artificial short-term cultures. We think that these are hard to kill. We think that these are relatively easy to kill. So what we're going to do now, what we're doing in collaboration with Alberto Bosque, is to mix these two together in a dish from the same person. Then we add our different shock and kill approaches and see if we can kill the latency model and see if we can kill the real model. And how do we know which one we've killed? The latency model is all with the virus NL43, a clone, and the patient reservoir is a different virus. So we can just sequence what grows out in these viral growth assays and say, did we reduce the model reservoir or did we reduce the real patient reservoir? And these data are a bit preliminary. We're doing more of these experiments, but I think they look encouraging enough to show. So with these all mixed together, if we look at the viral outgrowth assays, here we haven't separated the model and the real reservoir. Now, for the first time, one of the first times, we see a very slight but significant decrease just by adding the latency reversing agent and the CTL. And again, we see an enhanced reduction where we add the BCL2 inhibitor. But what does this mean? Have we killed the model system or have we killed the actual real reservoir? So when we overlay this with our sequencing, we can ask which viruses were reduced. And in the model system, so this is on a log scale, the model system, we do see a 61% reduction in the amount of virus with L43 in it, which was significant in contrast to the natural reservoir under the same conditions, which had a non-significant increase. However, for both of them, again, where we added the BCL2 inhibitor, there was a much more striking decrease in this reservoir. So the results from this initial experiment is that the BCL2 inhibitor did enhance elimination of cells from the model, but we feel that it's actually unlocking the ability to induce some amount of elimination of ex vivo infected CD4 cells, even though for many, many other experiments, we weren't seeing any change in this viral outgrowth assay. With the BCL2 inhibitor, we can start to see some changes. So conclusions, combinations of potent latency-reversing agents with cytotoxic T-cells drove reductions in HIV DNA, which we think are consistent with elimination of some of the defective proviruses, which ties into what we showed with the XIII, that some of them can be expressed. But these were not associated with significant reductions in the infectious reservoir measured by this kind of gold standard QVOA assay. We went to some lengths to demonstrate that this was not attributable to immune escape or to immune dysfunction. And how we know that is we can take the virus from the QVOA, infect activated cells, and that same CTL clone can kill those cells. But we have seen in the experiment I showed and in others, CTL-mediated reductions in primary cell models. So there does seem to be something different about these real natural reservoirs, these cells that are taken directly from people. And what is the mechanism of this? I mean, we have one clue to follow with the BCL2 inhibitor, but we really don't have the full mechanism. But when we added this inhibitor, finally we were able to see substantial reductions in the infectious reservoir, at least under these controlled ex vivo conditions. So this idea that maybe we need prime shock and kill to actually achieve these reductions. So take-home messages, or just put another way, we initially thought that viral reactivation, or not we, but the field at first thought, if you just wake up latently infected cells, they'll die. Then, in the landmark paper from Liang Shan, it was shown that this was not enough to reduce the infectious reservoir from patient CD4 cells. But if you combine latency-reversing cells with models of latency with CTLs, you could reduce them. But the question that we are trying to go back to is, does this actually work with patient CD4 cells, with these real natural reservoirs? And in our hands, we were surprised to find that it consistently doesn't. Even strong latency-reversing agents and potent cytotoxic T-cells were not able to reduce the infectious reservoirs. So the question is, do reservoir-harboring cells, which, again, I keep making the point, but they've survived for a long time in these patients, and are often clonally expanded. That's something that the field is learning. There's often divisions of a single infected cell, suggesting it has some particular fitness aspects. Do they have some intrinsic resistance to immune clearance? And what are the mechanisms? So is it true that these cells are always hiding through a mechanism of latency in vivo? Or maybe they are seen by the immune system from time to time, but not efficiently cleared for whatever reason. So I will leave us with that provocative question and thank John Huang in my lab, who did most of this work, along with Yanqin Ren, also our collaborators, Doug Nixon, Camilla, and Esper Alberto, and of course the clinical participants. Again, we're highly dependent on these leukapheresis samples, and our clinicians and participants are terrific with this. And with that, I will end and take any questions.
It's open. So, great talk. I was wondering, I actually have two questions. First one is, have you checked the levels of expression of BCL2 in these queen B T cells, and the other T-cell, the other infected T-cells that are just the defective provirus. And you think, if yes, you think that if that cells express more levels of BCL2, which is an anti-apoptotic protein, that could explain why these reservoirs live so long since they are anti-apoptotic protein. And
sure. So thank you for the question. So I will also use that opportunity to make the point that there are many other types of viruses that are known to modulate the BCL2 pathway as an immune evasion mechanism. I don't know any precise mechanism by which HIV would modulate that, but one of the challenges in this field always is that directly observing these cells, these 1 million cells from people living with HIV is very, very hard. So I haven't been able to ask the question, and I don't know of others who have asked the question, are survival factors such as BCL2 preferentially expressed in these real reservoirs? I think I know of several labs that are trying to come up with methods to isolate those cells ex vivo. So I'm watching with interest to see what they find. But at the moment, I can only speculate and say that if we inhibit BCL2, it seems to help. So maybe it's something as direct as that.
So, and you have any thoughts that why the whole genome is required? Because you have cells that are infected with defective virus, and you didn't see the same phenotype. So, do you have any thoughts why the whole genome is required?
Yeah, another great question. So again, I have thoughts, but not data. So one thought could be it's something about some viral factor that we just don't know yet, which is missing from defective viruses. That's possible. Another thought is that there's a number of different immunoevasion factors at play, right? So intact proviruses will also have intact NEF, which will downregulate MHC1, whereas many defective proviruses don't have NEF. So that's a bit different from the BCL2 story, but if we have a number of complex factors interacting, so maybe the intact proviruses aren't recognized as well by CD8 cells because of NEF, and there's some BCL2 or survival manipulation in these infected cells, that kind of convergence of factors might give this survival advantage to the ones with intact proviruses. But it's something that, I mean, these results were really unexpected, and now that's what we're really trying to drill down into is the mechanisms, but we just don't know at this point.
Any other question?
It was really a great story that you're telling, and it's really interesting. And your suggestion that having survived as clones for a long time might identify something about those cells that makes them resistant. I was wondering on the other side whether or not the fresh infection system had any sort of activation, manipulation of those targets to make them tastier to CTLs, just to look the other side of the control, like activation with something or...
Yeah, that's a great question, and I think at one point that would have been a really important distinction because our latency reversing agents were things like bryostatin or IL-15, which are very different from how we're treating our in vitro models, which involve anti-CD3, CD28 stimulation. So maybe that anti-CD3, CD28 stimulation was priming those targets for elimination. But by the end, where we've got to, our LRAs, latency reversing agents, are also CD3, CD28. So the durations of treatment are not identical. There's some things that are a bit different, but the cells are generally being treated in in fairly similar ways. One difference, again, from the 2012 immunity paper was it was a NEF-deleted virus, so that also certainly helps the targets be tastier. Shoot, I was going to make another point, but I forgot. Maybe it'll come back to me.
Well, despite your memory lapse, it was a great talk.
Thank you. Oh, sorry. One other point I was going to say. So the TL side, I think, is obviously what we're focused on. But one thing that I also want to highlight and not get lost in the details is that also in these primary cell latency models, strong LRAs alone really reduce the reservoir. If you add anti-CD3, CD28, if you add bryostatin, if you add PMA autonomycin to the Bosque model, other models, and then do a QVOA after, you've really wiped out the infectious reservoir. So even if for some reason our CTLs aren't working the way that we think, which I don't think is the case, but maybe who knows? It's weird that hitting these with such a hard latency reversal hammer and for 4 days and then putting them into viral outgrowth assays isn't resulting in any reduction in the reservoir, which is different from those model systems.